Hello and welcome to part 3 of this video series on homeostasis and the kidneys for A-level biology. Big thanks again to @era.anatomy for the backdrops that are going to be going on from now on, and a big thank you to the students who've been requesting this topic, particularly Amelia, sorry it's so late. So, this one, I would say, is probably the number one area that students get confused by, is what goes on in the loop of Henle. It is very different and additional to what you did at GCSE, but I think the number one thing that you can get right to make it easier is the language, okay? So we need to be using relative terms here. If you're just wanting to say, the sodium goes, or the chlorine goes, or the ions move, well, unfortunately, biology isn't about absolutes. Biology is about changes in relative concentrations of things. So I want to see words like more, less, higher, lower, greater, smaller in exam answers. You know, talking about gradients and differences, not all of it. Not this thing goes, all the water goes over there, so there's no water over here. Because, of course, biological systems are predominantly water, and it's only ever percentages of that water that are moving around, similarly with percentages of ions, all that kind of stuff. A former student of mine who's now at Aberdeen University studying medicine talked about, when she did a talk for our students, talked about the idea she had no idea, hadn't really appreciated till her medical degree, how much of biology and anatomy is about water potential, how much of it's about the relative movement of ions and water. And that's very, very true in this topic. Now, the good words to use here, and I think this is possibly the number one area where students get mixed up, are concentration and volume, okay? If you're using the word amount, right, amount as absolute amount, the amount of this, the amount of that, you are going to lose marks and you are going to confuse yourself, okay? So you've got to use the correct terms. What is the concentration of the solution? What is the volume of the solution? Okay, and let me explain a little bit more about why that causes confusion. I think it's fundamentally because students don't truly internalize the ways of changing concentrations of solutions. So here I've drawn a very simple sort of container of water and some dissolved thing, be it sugar, ions, salts, doesn't matter. Okay, so this is our solute and this is our solvent, C Year 8 chemistry. Now there's a number of different ways of changing the concentration of this liquid, okay? Now the obvious ones, okay, add more dissolved stuff. So you add a bit more dissolved stuff from here to over there, it becomes a more concentrated solution. Note the blue box is the same, the volume of water is the same, but the volume of dissolved stuff, the solute, is higher. Therefore that is a more concentrated solution than that, okay? Of course, the other way is to just add more water, okay? Add more water in. So we've got the same amount of dissolved stuff, the same amount of solutes in there, but by adding a bit more water, we've made it a less concentrated solution. We've dropped the concentration. Of course, this here would have a lower water potential, C Year 12, than that, okay? So I am going to assume that you know about osmosis and you know what a water potential is for the remainder of this video, okay? So we've got add more dissolved stuff, your concentration goes up. Add more water, your concentration goes down. But then, of course, there are the other things that the kidney is doing, right? Which is a different way to alter the concentrations of the solution, okay? You could selectively take away some of this dissolved stuff, right? Typically ions or sugar or whatever it might be. So you might remember the last video, we talked about the selective reabsorption of amino acids and glucose. That is going to raise the water potential of what's left because there's less dissolved stuff in it, so the filtrate becomes a higher water potential even though it's a slightly smaller volume, okay? Because in comparison to the water, there is less dissolved stuff. Right? So this is our relative concentration. We haven't really changed the volume significantly, but because we've taken dissolved stuff out, we've raised that water potential, right? And we've lowered the concentration, okay? The other way, of course, would be to go the opposite way. If we were going to take water away, right? Well, then we wouldn't have to change the amount of, or change the volume here, right? We have to change the volume of dissolved stuff, but if we start taking water away, then of course we become a more concentrated solution. We lower that water potential, okay? We've not added in any more dissolved stuff, but actually what we're doing is increasing the concentration by taking water away, okay? So So we can add or take water, and we can add or take solutes, okay? Now, this is separate from there is more of, right? The amount is higher, the amount is lower, right? Be specific when you're answering these questions. Concentrations, volumes, relative to one another. Words that ends with ER, greater, higher, lower, okay? This is what we want. So, we are now here, osmoregulation, we're in the loop of Henle. So we've been through ultrafiltration, we've been through selective reabsorption, we're now going to go into osmoregulation, right? The regulation of water down in the loop of Henle. So, if we look specifically, right, we're basically looking at a simpler diagram here. You've had the afferent arteriole come in, be ultrafiltered, all the filtrates gone into the Bowman's capsule, the filtrates come through into the proximal convoluted tubule. You've had that co-transport away of glucose, sodium, amino acids, all that kind of stuff. And what you're left with is generally a mixture of salt, dissolved ions, and water. Okay, and obviously the waste products, urea, that you wanted to get rid of. So, I'm going to zoom in just on this little bit, and we're going to label this. So because selective reabsorption has just happened, okay, we've got a relatively high water potential as the filtrate comes into the loop of Henle. Now the descending bit, the bit that goes down, the descending arm of the loop of Henle here, is impermeable to ions but permeable to water. Okay, now out here we've got a lower water potential in the tissue surrounding the loop of Henle, largely because we've been selectively reabsorbing a load of dissolved stuff. Okay, so the water potential here is lower than it is in the filtrate. That's going to lead to osmosis. Okay, that's going to lead to water moving out of the loop of Henle into the surrounding tissues, and that's going to change the water potential. Here is going to become more dilute, and here is going to become more concentrated. So as we go down, right, water diffusing out into the tissue and eventually into the blood, what's going to happen is we're going to end up with a higher water potential down here, purer water. We're adding water in, so we're making the solution more dilute. And because water's coming out of the loop of Henle, that's going to end up being a lower water potential. Okay? So that descending arm makes a more and more and more concentrated solution, but we still haven't got enough of the stuff out of the filtrate to call it urine yet. We wouldn't want to lose the filtrate that's down the bottom here, because we wouldn't be able to survive very long, right? And by retaining enough water to survive. So we need more mechanisms, more multipliers to make this happen more effectively. So as the filtrate travels back up, the ascending is impermeable to water, but it is permeable to ions, okay? So out come the ions. And of course, what they do is they lower the water potential of the tissue around the loop of Henle. And the reason it's a loop, okay, and these are close to each other, is of course this tissue here that's right next to the ascending limb, the water potential of this tissue is going to get lower, which is going to speed up the osmosis of the new filtrate that's coming in here, okay? So we've got this kind of multiplier effect. Ions coming out here, lowering the water potential around the descending limb and making water come out faster by osmosis. So by the time it gets to the bottom, you've got quite a concentrated solution, and then that solution goes up and it in turn donates its ions to lower the water potential here to bring more water out by osmosis, okay? You can make some synoptic links here with countercurrent flow in fish, and the idea that in biology, when things flow in opposite directions to one another, you do get kind of multiplier effects of how they work. Same is true of heat in a duck's foot, right? Heat in the end of your nose with blood vessels with warm blood coming in, and that's heating up the cold blood that's coming back from the end of your nose or from a duck's feet. This kind of idea in biology is a theme that's repeated in a number of ways, and this is just one example, okay? So one more time, very nice, pure, high water potential filtrate comes in. The tissue is a lower water potential because of all the dissolved stuff in it. Water moves by osmosis. As it goes down, this becomes more concentrated and decreases in volume, right? But it's still got all those ions. Because all the water's come out here, it's a higher water potential, right? As the filtrate flows back up again, you end up with permeability to ions. So out come the ions, lowering the water potential and multiplying the effect of that osmosis, okay? And that effectively is what goes on in the loop of Henle, okay? So moving on, right? Just to put our little cups back on that we did in the previous slide, up here you've got a fixed amount of dissolved stuff and lots of water, so high water potential. As you go down, the dissolved stuff isn't changing, but the water is decreasing. Therefore, the solution is becoming more concentrated. Okay? It's a basic concept, but one that's absolutely vital to understand this. As we go back round again, as you can see here, the water is now fixed. So the water doesn't change up to there, but the amount of dissolved stuff does decrease. Okay? So therefore, the remaining water here becomes more dilute, even though it's a significantly lower volume than it was to start with. Okay? So this is why we want to talk about concentrations and volumes, not just amounts, and relative to one another. So, moving on. If we put that in a little table, in the descending arm, the water comes out, the ions stay, the water potential goes down and the ion concentration goes up relative to the water. In the ascending arm, the ions come out, the water stays, the water potential goes up and the ion concentration goes down. Then into our distal convoluted tubule, which is our bit for osmoregulation and ADH, we get a low volume, right, of low volume of higher water potential filtrate. Okay? And on that note, right, I shall leave it there. Next time, ADH and osmoregulation in the distal convoluted tubule. Thank you very much.